Good afternoon, everybody. My name is David Bott, and I have the pleasure of chairing this afternoon's SCItalk on the quest for effective dementia treatments. So in this talk, Ruth is going to talk about what dementia is, how difficult it is to understand the underlying mechanisms, and therefore to create new medicines to deal with it. She's been involved for a long while as a neuroscientist, and she's also seen lots of other people do all sorts of things over the years, and she's going to describe that and explain it to us. And we're going to know a lot more by the end of the next hour. As I say, my name is David Bott. I'm chair for this. I don't nothing like that because I've got a lockdown mentality these days. And the Q&A will be coordinated by Will, who you will hear later. Ruth has been an academic in business and in government. I first met her just over 10 years ago, where she was in Pfizer and was talking about neuroscience, and we wanted her to work with the TSB. She liked it so much, she ended up running what became Innovate UK, but she's maintained her career as a scientist and as a communicator about science. So it's more than just being a scientist and talking to other scientists. Ruth has a very great skill in talking to normal people about science in a way that's understandable. As I said, when I first met her, she was at Pfizer in Sandwich. She moved on and helped develop all sorts of things, and then retired from Innovate UK a few years ago, and now is a venture partner with SV Health Investors and does a whole bunch of stuff helping small companies, has one herself, and just, you know, is an all-round good guy in the scheme of things. So with that, I would like to turn over to Ruth, and I'm looking forward to this, Ruth, so good luck.
Thank you very much. Hopefully I'm sharing my webcam and you can see my home office, and I'm also going to share my screen so that you can see my slides. And I'd just like to say, bear with me, because I'm not so used to doing this and it might not be that easy. Let me see. Can you now see that? David, please answer.
Yeah, I can see it.
Okay, great. So thank you very much, David and SCItalks, for the invitation to give this. Now, normally I would much prefer to do a face-to-face lecture, and you know, so there'll be much less interaction than I would ideally like. However, there will actually be some audience participation, so be prepared. You will be asked to fill out a form at some stage during the lecture, and hopefully we'll get a little bit of feedback from that. So today I'm talking to you as somebody who spent over 20 years as a neuroscientist in the pharmaceutical industry, trying to invent new medicines for brain disorders. And in putting this talk together, I'd like to recognise two of my closest colleagues who I've worked with for decades in different pharma companies. So at the top there is David Reynolds, who used to be the CSO of Alzheimer's Research UK, and I should disclose that I'm a trustee of that charity, and it's a wonderful charity. And the second person I want to really acknowledge for their effort and help is Paul Whiting, who is the head of the Drug Discovery Institute at UCL, working on dementia. And I know both of them well enough to shamelessly borrow their slides and some of the ideas that they've had, and they've been big contributors to my presentation. I'd also like to acknowledge the Dementia Discovery Fund, of which more later. That's the investment organisation that's part of SV Health Investors, and it is covered by the Financial Conduct Authority. So I just want to be clear that I'm talking to you today as a scientist, so don't be making any financial decisions based on the information and anything I say in my talk. So the outline of my talk, this is what I intend to cover: a little bit about dementia, and then I'll explain the two major long-standing theories on what causes dementia, these two proteins, amyloid and tau, and the scientists who appreciate a bit of friendly rivalry. And so Alzheimer's disease researchers have been divided into the BAPtists and the Tauists for a couple of decades. I'll take you through how medicines are developed, and medicines addressing both of those mechanisms are now in clinical trials. And there's been a lot more research done since then, and more funding. And I will tell you something about how things have changed. And then, as I am a very positive person, I'll end with my personal view on why we should be optimistic about medicines for dementia. So dementia covers a variety of diseases and symptoms, not just memory loss, but a lot of other things. There are a whole lot of language problems, disorientation, mood swings, behavioural issues, and the prevalence is increasing. Many of you will have had or do have family members who are suffering from Alzheimer's disease or other dementias. The prevalence has been growing in the population, so now 10 to 30% of people over 65 experience some symptoms, and it's actually very rare that Alzheimer's disease is inherited. So very rare that you would have it as an autosomal dominant disease and that you would be able to pass it on to your children. Less than 1% of Alzheimer's is of that type. The Alzheimer's Research UK recently carried out a survey, and they found that only 23% of the people they surveyed thought that dementia was caused by a brain disease. It is absolutely not a natural consequence of aging. The incidence of dementia has been increasing at the moment. We're coming up to 1 million people living with dementia in the UK, and by 2025 it'll be over 1 million, and by 2040 over 1.6 million. So what is there to help us with fighting this disease? I start with this picture from the Daily Express that coffee is a treatment for Alzheimer's disease. Dementia is a much feared disease, and there is a lot of absolute nonsense written about it, of which this is one example, chocolate is another example, spicy diet beating dementia is another example. The high quality science written about dementia looks more like this: drugs targeting E1F2 alpha or TREM2 mutations. For most people, these scientific articles are hard to understand and really, really difficult for the non-expert to conceptualize. And so I'm going to do my best today to bridge the two, to bridge the science and the commonly understood tenets of dementia. So first of all, there are many types of dementia. It's an umbrella term, and it's a description originally based on post-mortem pathology. You can see here, I hope you can see here, can you see the pointer? Yes, no?
Yes.
Yes, now.
Yes, you can now. Okay, so you can see here that more than half of people with dementia have the Alzheimer's type of dementia, but vascular dementia is also very important, with changes in the vasculature and micro strokes, for example. And then pathology describes other forms, such as dementia with Lewy bodies, which is very common, frontotemporal dementia that starts with the executive function at the front of the brain. And actually, there is a Parkinson's type of dementia that is based on loss of a different brain part specific to Parkinson's disease. And then there are other types of dementias as well, much rarer. So it is a disease of neurodegeneration, the loss of brain cells, mostly neurons. And if we have a look at how that appears in the human brain, at the top there you can see the cross section of a normal human brain, and in the bottom section a late stage Alzheimer's disease where the brain as a whole appears shrunken. So there's extreme shrinkage in the cortex, and the ventricles, those black circles in the middle which normally circulate the fluid that goes around the brain, look much larger. There's a loss in the hippocampus, which is important for memory. And all in all, by the time somebody is in the late stage of Alzheimer's disease, they have lost about 150 grams of brain tissue. And that's about the size of a small orange. Alzheimer's disease takes decades to develop, although some forms like frontotemporal dementia are more accelerated. So the challenge really is obvious: how to stop the brain degenerating. And Alzheimer's disease and other types of dementia are the hardest disorders to work on. Arguably, they're harder than cancer, because in cancer, you're trying to stop cells from dividing out of control. In neurodegeneration, you're trying to stop neurons from dying. And neurons aren't like skin or liver cells. Once they're made, they pretty much don't divide or grow during life. So it's virtually impossible to bring them back. The challenge then is how to stop them from dying. There are actually some drugs already available that are currently used to treat Alzheimer's disease. And in the graph here, you can see that cognitive function does decrease slightly with age. In normal aging individuals, the green line on the top, there is some loss of cognitive function. But in people who have dementia, in the red line, you can see they cross into a point of mild cognitive impairment where they need additional support, and then on into frank dementia where they need much more active care. And the drugs that are available at the moment, the three drugs most commonly used, Exelon, Aricept and Namenda, they work just at that boundary between cognitive impairment and frank dementia. And what they do is they boost the ability of the neurons that are still alive to communicate a little bit better. They pep up the transmission of signals between brain cells. They don't stop neurons from dying, they just allow those that remain to work a little bit better. And so their effects are relatively short-lived and they don't actually change the number of brain cells. These drugs were first discovered around about the 1980s and 90s, actually worked a little bit on Aricept myself. And since then, there have really essentially been no new medicines for dementia. So in order to make drugs that can prevent the disease, we have to understand what goes wrong. And the story of what goes wrong, the quest to understand that, started over 100 years ago. So this on the left here is Alois Alzheimer. He was the clinician that worked in Germany at the Institute for the Mentally Ill and Epileptics in Frankfurt. And this is his really important patient called Auguste Dieter. She died in 1906. And Alzheimer asked for her brain so that he could look at the pathology of what went wrong. So Auguste had the full set of symptoms of dementia. She had memory and speech problems. She actually also had hallucinations and then some of the unpredictable behavior that you would see and disorientation. Her brains were sliced and looked at very carefully, and what you can see on the bottom of this slide is Alzheimer's handmade drawings of what he could see in the brain. So he could see large sticky clumps, which were very common throughout the brain, and we call those plaques. And he could see much smaller diffuse tangles, which are present inside the neuron. So the drawing on the left is of the tangles, and then the top right of the drawing on the right is a plaque. Now, Auguste was only 55 when she died, so quite young for a dementia patient. And as it turns out, based on genetic analysis on these sections, a century later, it transpires she is one of the rare, less than 1% of patients that have an inherited form of the disease. So the pathology of dementia is now quite well understood. And let me just see, in this slide, you can see not a handmade drawing, but a stain of the pathology from a control person and somebody with Alzheimer's disease. And we now understand that in a control patient, there are really no plaques, particularly in young people. But in Alzheimer's disease, you can see here the amyloid plaques, and here's one close up, made of a protein called amyloid. The tangles that were described by Alzheimer's, you can also see not present in young control patients, but more diffuse and present in an Alzheimer's patient. And you can see that they are forming a swelling of the ends of neurons, and you can see these proliferations of tangles in the slide here. So understanding what causes plaques and what causes tangles has been the life's work of many teams across the world. But I'm just going to highlight two people specifically. So in the top there is John Hardy, who is a professor at University College London. And his team did the seminal work that found some rare patients who have a genetic mutation in a protein called amyloid precursor protein. And that protein is a much larger protein from which an extract is produced that causes the plaques. And for that work, his team won the science equivalent of the Oscars, the 2016 Breakthrough Prize in Life Sciences. On the bottom there is another quite famous individual, Michel Godert, and his team discovered that tangles are made of tau. And he also found a rare patient that has a mutation in a gene that produces tau called the MAPT gene, and that causes frontotemporal dementia. So these two really famous and wonderful scientists, along with two others of their colleagues, Christian Haas and Bart de Strooper, were awarded the Brain Prize in 2018, and fantastic achievements. Their collective discoveries provided really the evidence that amyloid and tau were, and I say that, they provided evidence that they were the cause of dementia, but I think we now would say they're not, they're necessary, but they may not be sufficient to cause dementia. So more of the story to unfold. What do we know about tau versus amyloid disease? To recap, tau is involved in transporting molecules along the nerve. It's the railway system, if you like, of the neuron. It's not hard to imagine how stopping the transport of nutrients or building materials and information along the neuron could damage the cell and ultimately cause death. Interestingly, with tau, the formation of those tau aggregates correlates with the onset of disease and the region involved in the brain disturbances. So tau makes the fibrillary tangles, it correlates with clinical onset, and it's in the brain region that drives the symptoms. So the front part of the brain, the executive temporal lobes, are involved in decision making. When you have tau aggregates there, it's really hard for people to think, to plan, and their behavior becomes quite erratic. For amyloid, surprisingly, we still don't know exactly what amyloid does in the brain, although many more genes have been found associated with dementia, not just APP, John Hardy's discovery, but actually a few others, and one of those that Augustita had. So she had an amyloid type of dementia. For amyloid, these make plaques outside the brain, all the way across the brain, and actually the plaques occur decades before clinical onset. So not reassuring, but if you're in your 60s as I am, the chances are you've already got plenty of amyloid plaques, but they're not specific to the area involved in symptoms, and for many, many years they appear to be relatively harmless. Let's talk then about how to intervene. We need to know and understand what the biology is, and we need to know and understand how molecules could interfere with that. So John Hardy's discovery led pharma and biotech companies to focus on a very small part of this amyloid precursor protein. And if you can see here in the red box is this little yellow line, and this is the fragment of the amyloid protein that is cut out, made into little monomers, which then aggregate. They stick together. When they stick together, they become toxic. They kill synapses, and when those toxic small oligomers stick together, they become much less toxic and form those major plaques outside the brain. They are the hallmarks of the disease, and there are many ways actually to think about intervening in that pathway. First of all, we could consider the enzymes that form that beta-amyloid peptide, and there are two. One is called BASE and one is called gamma secretase. Maybe if we were able to block those enzymes, we might stop the toxic monomers from being made. The other approach we might take is to try and get rid of the amyloid once it's made. So use antibodies to bind to amyloid and label it for destruction by the immune system. And all of those approaches have been taken. So BASE inhibitors have been pursued, as have gamma secretase inhibitors or modulators, so that they cut in a different place. And antibodies that bind to amyloid and stimulate clearance mechanisms have also been used. And actually quite a lot of drugs have now been developed, and I am not going to pronounce all of these very difficult names. Suffice to say that there are drugs that inhibit BASE, gamma secretase, and antibodies that bind to the aggregated amyloid that have made their way into clinical trials. How about the tau hypothesis? What have the Taoists to say about making drugs? This is what happens in their hypothesis. First of all, the tau protein is made inside cells. It forms that transport mechanism, and the proteins aggregate again and produce really small oligomers that are not helpful in the neuron. What's different here, though, is in tau, those tau toxic species are released from one cell and transported to the next. So they spread from one neuron to the next, and that causes tau toxicity to spread throughout the brain, starting in the frontal parts of the brain, but spreading in later stages of the disease into much broader regions. So is it possible to reduce tau production or stop it aggregating? Is it possible to impair the toxicity of tau in the brain? How would you go about doing that? So similarly, you'll notice that as for the amyloid hypothesis, many approaches have been taken. Here, most of the drugs are described in the yellow boxes are a combination of letters and numbers rather than names. That's because these are in the earlier stages of development, so the molecules have a code that refers to the company that's making them, such as Lilly or Roche or AstraZeneca, and only when they get into later stages of clinical trials do they formally get a name. How then are those drugs developed? Where do they come from? How do scientists work through making a drug? Making medicines is a long and very expensive process. It starts with a light bulb moment, an idea on how to do that. So do we reduce amyloid in the brain? Do we reduce tau in the brain? We have to pinpoint a way of doing it, would be the second stage. Shall we try beta-secretase, or shall we try an antibody? If it's an enzyme or receptor is the drug target, then we have to find the compound that acts on that molecule and refine it so it has the best properties to form a new drug. Is it suitable for giving it orally? Is it potent enough to be specific for just that enzyme in the brain? Is it broken down too quickly so as not to be useful? And then the next step is to try and show that a putative molecule does something useful that would support its development clinically. So quite often molecules would be tested in mouse models of dementia, where the mutations that arise in the patients with rare mutations are replicated in mice, and we look to see if the molecule can work. And only if it gets to that point can we then think of taking it forward into clinical trials. But which drug to pick? How do you know which is the right target? It's like a quest for the Holy Grail, and I hope you can hear this. And I think we know what happens next, so we won't watch any more of that. Let's continue with what happens when we are trying to develop drugs. So having selected our target, what we believe to be the Holy Grail, selected the molecule that we believe to be the Holy Grail that will work, and hopefully it works in animals at step number 5, then we can move into clinical trials and explore whether a drug is safe and effective in people. And in order to run clinical trials, there are actually many stages. So first of all, a drug is tested in healthy volunteers to know that it's safe, it's tolerable, to understand something about the pharmacokinetics, what dose we need to give. After that, phase 2 is understanding whether the drug works, and then moving to phase 3, how do we compare it with other drugs and give it to patients more broadly? And then once the drug is registered and approved, there is plenty of post-marketing surveillance that needs to go on to understand a little bit more in detail about the medicine. That takes more than 15 years, and for most drugs costs about £1 billion. For drugs to treat dementia, because the clinical trials have to be so long and take up so many patients, that cost is more like 5 billion for any one drug and requires a large team of scientists and clinicians. So getting a new drug through research and into clinical trials is fraught with new challenges. It takes experience and luck to pick the right path through many obstacles. It is a leap of faith to move into clinical trials. There are many bumps in the road. Sometimes it's necessary to take a step backwards before you can move forwards. And there are many tight timelines to be met. And all under an incredible weight of expectations from patients, from a chief executive officer who signed the budget, and the company's shareholders who are really anxious to see a positive outcome. But it is the only way to find new medicines. So how successful have we been? Testing drugs in Alzheimer's disease has been challenging. And in this figure here, I'm showing you where we were about 10 years ago. This was the number of medicines, ideas that were preclinical. This is the number that were in phase 1 clinical trials in early volunteers. These are the different molecules that were being tested in patients. And these were the molecules that were in phase 3. Now, some of these are based on what we understand about the mechanisms of the disease, like bapineuzumab, like solanezumab, and some of them were ideas of drugs that already existed that might also be useful in Alzheimer's disease, like rosiglitazone, which is more commonly used in diabetes, and statins that are more commonly used for keeping your cholesterol under control. So many ideas. And how many of these so far have been successful? The answer is a disappointing 0. And the clinical trials have been long and very expensive. So expensive, in fact, that some pharma companies have joined forces to be able to afford them. Current examples would include Lilly and AstraZeneca, who have been co-funding the development of a BACE inhibitor, and Eisai and Biogen have been working together on an anti-amyloid antibody. And some companies, such as Pfizer, have actually got to the point where they've chosen not to work internally on dementia at all. This was the situation in 2012. It is disappointing. And for the amyloid hypothesis in general, the gamma secretase inhibitors haven't worked. They had many side effects because the enzyme's present throughout the body, and drugs affecting gamma secretase not safe enough to be used. The BACE inhibitor, the other enzyme that produces the amyloid fragment, actually made patients worse. And to Merck's credit, the company that had the leading molecule, as soon as they saw that information, they shared it with their competitors who had similar drugs in development because they wanted to minimize the risk to patients. For the antibody approaches, most of the jury is currently still out. There is a lot of work still to be done. Now then, the late stage failures and the reticence of pharma companies to continue investing had caused really a bit of a wake-up call for funders and for scientists and for government as well. With an ageing population and the incidence of dementia rising across the world, the economic as well as the healthcare consequences loomed large. And actually, it was David Cameron who took the lead at the World Dementia Council in 2003, and he shone a light on the need for more funding for both basic research and for the development of new therapeutics. It was time to look beyond tau and beta-amyloid. It was time to try new approaches. So funders of all flavours actually pulled together. The Medical Research Council, along with the charities Alzheimer's Research UK and the Alzheimer's Society, formed the UK Dementia Research Institute, which has its hub at UCL and includes 5 other universities across the UK. and the Dementia Discovery Fund was formed. And this is a patient capital investment fund, which has had contributions from the British Business Bank, the Department of Health, 7 pharmaceutical companies, Alzheimer's Research UK, the Gates Foundation, and actually many others. And collectively, that fund is in London and Boston. It's based on investing the £250 million they raised in new companies to take forward new approaches to dementia. So what has that additional more than £500 million in investment done to help us in our understanding of dementia? First of all, let's look at the research. Now, research goes on across the world all of the time, but it has been boosted in dementia in the last decade. Much of the new research has focused on trying to understand neurodegeneration beyond amyloid and tau. And here's a selection of the latest discoveries. There are very few genes, as I keep saying, that are inherited and dominant. But there are many, many genes out of the 25,000 genes that constitute our biology. About 100 or so also contribute in some small way to the risk of developing Alzheimer's disease. The top of my table here is ApoE4, which you may have heard of. Now, if you have 2 copies of the ApoE4 gene, then you will have an 8 to 10-fold higher chance of developing Alzheimer's disease than if you have 2 copies of ApoE2. Many of the genes that have recently been discovered to be involved in the risk of developing Alzheimer's disease are not even in neurons. You'll see some of them there are in astrocytes, some of them are in what are called microglia, which is the immune cells of the brain. There are many new ideas opening up for how we might treat the disorder. There are more chalices to choose from and a much more sophisticated understanding that dementia isn't either tau or amyloid. As we understand more about dementia, we learn that the risk of getting it sits across many other genes, and actually even a small part of that risk, maybe 20%, is modifiable. Modifiable by good health in midlife, by not smoking, by taking exercise, by keeping your blood pressure and diabetes under control. All of that can help to manage your risk of getting dementia, but it absolutely can't stop it. If your biology is directed towards you getting dementia, we still will need drugs to be able to treat that. Let's look at what the Dementia Discovery Fund has done. So far, this fund has invested in 16 new companies, and here's a snapshot of them. I don't expect you to look at them all, but they have different mechanisms, all have new approaches, not working on tau or amyloid, and I think this is a bright future for research and the creation of new drugs. It would be remiss of me not to mention just one though, and that is the company Astronautics, second down on the left there. And the reason I mention that is it's not based on neurons, it's based on astrocytes, the support cells of the brain, the civil servants, if you liked, the ministerial neurons. And my colleagues, David Reynolds and Paul Whiting, who I mentioned right at the very beginning of this talk, are co-founders with me in this company. And one of the many hats I wear is to be their chair. It is a really exciting time for new medicines in dementia. So here's our poll. Here's my question for you. Based on everything I've said, the new biology, where we were 10 years ago, how many clinical trials do you think there are in dementia now? And I'm giving you 4 options, and the poll is open. And included in clinical trials, I would ask you to consider trials that are looking at the natural history of the disease, trials that are looking at developing new biomarkers, new processes, new ways of monitoring the disease, those that are continuing to work on tau and amyloid, and those that are involved in maybe repurposing existing medicines and in some of the new methods, some of the new science that I've described. So that's your choice. I will wait to find out what the answer is. You need to vote. Have you finished voting? And can we get what the answer is?
Results are showing now, Ruth.
I can't see them.
So 0 to 50 is 26%.
Yep.
50 to 100 is 23%. 100 to 1,000 is 33%. And over 1,000 is 18%.
Well, that's interesting. There are actually over 2,000 clinical trials going on for dementia. So a fairly evenly spread vote, but actually I think it's a very positive story. And I'm just breaking down here. You can see the different types of clinical trials that were being run up until the end of last year. And what is interesting, for me at least, is if you look on the right, the ability to get as far as clinical trials depends on the amount of science that has been done, on the number of scientific publications. So amyloid was the first hypothesis to be developed, and 47,000 publications and the largest proportion of clinical trials. Tau in grey there, 15,000 publications, and 12% of clinical trials are looking at the Tau hypothesis. ApoE, the more recent gene to be discovered, a lot of publications, but we still don't really know what that protein does, and it hasn't made its way into clinical trials yet. And all of the other segments of this diagram are made up of some of the newer hypotheses looking at energy provision or disorders in the vasculature, and particularly in inflammation. So a really positive time. And let's just summarize with what we know now. So most drugs based on the amyloid hypothesis have been unsuccessful. They have failed. There are some hints of benefit from two of the best antibodies. So these are solanezumab, which failed in clinical trials, but in the least severe patients, earliest in the disease, there were some hints of benefit. And aducanumab, which is still in clinical trials after some inconsistent data, that continues, and we look forward to seeing how Biogen's drug fares in the studies it's currently undergoing. The Tau hypothesis, most of those didn't do well. Anything looking at transport in the neuron failed. Most of them were toxic. Most of the effort now is on antibodies, and we will hear more about those in the next year or two. Research is creating new ideas all the time. You can't do too much research. So some of the new ideas are how the brain manages aggregated and disordered proteins. How does it deal with things that are improperly made or aren't transported properly? So first of all, the brain has a glymphatic system. Fluid washes through the brain, and most of the toxic substances are cleared during slow wave sleep. So there's two areas to look at there. The brain has a very good immune modulation system, where it also removes toxic species and keeps anything that is not properly formed in check and removes it. So a lot of work now on immune modulation. And then managing protein degradation generally. There is an emerging theory that the reason that amyloid and tau are toxic is not because they are per se involved in neurodegeneration, it's because they are the least soluble proteins in the brain. So when the brain is under stress, when it is dehydrated, for example, or after a stroke or an injury, you're much more likely to get pH changes in the brain, to get disorders of solubility, and proteins that are barely soluble will be the first to become toxic, and that would include amyloid and tau. So current approaches are more likely to slow progression rather than halt or reverse the disease. And I think what you can see here is that combination therapies will undoubtedly be necessary. I haven't talked about some of the tools that have been developed to help us understand whether drugs are working, to help us stratify patients, to know which type of the disease you have. Some patients have tau problems, some have amyloid, some have both, some have neither. And imaging agents help us understand which drug should be matched to which patients and at what stage of the disease a patient might be. So if you can see neurofilament light in the blood, that is a protein only expressed in neurons in the brain, and it tells you that this patient is losing neurons. we have learned a huge amount about dementia. We have learned a huge amount about how drugs can be made to treat dementia, but we're not there yet. I'm just showing some useful sources of information. I'd particularly highlight Alz Forum, which has all the most recent information. Alzheimer's Research UK, of course, is a great place to go for information and understand what is being done. And for more care type information, the Alzheimer's Society is very helpful. And I think that if we have time, this would be the point to show a video which I'm particularly fond of, which is from Alzheimer's Research UK. I find it pertinent and moving, and I hope you enjoy it. And that will end my talk. Thank you.
Thank you, Ruth. Imagine a virtual round of applause for an excellent presentation. You can't hear them, but they're there. Couple of questions from me before we open up to the floor. You sort of hinted at diagnosis towards the end there. I mean, is it possible to tell from physical attributes whether somebody has the early stages of dementia? I mean, even the different forms, or are we still into? I mean, I know they do the tests to work things out, but is there a physical test they can do yet?
Well, so interesting question. You can detect tau pathology. You can detect amyloid in the brains of people in a scanner, but that doesn't necessarily correlate with the disease. You can detect neurofilament light in the blood, quite a recent discovery actually, and that tells you if you're losing neurons. But actually, in my view, the most valuable way of knowing whether somebody has the disease and if they're progressing is not to measure physical things, but to measure the manifestation of the disease, which is the cognitive and behavioural ability that a person has. And actually, one of the other areas that I'm working on at the moment is trying to come up with a really good way that you can track the progression of somebody's disease, you know, preferably at home, not in a clinic, by getting them to do specific tasks that read out what different parts of the brain would normally do. So memory tests, not to train the brain to prevent the disease. I think that's actually, you know, a very, very small effect, but to monitor whether the patient is declining. And then it will be easier to see whether the drugs work. One of the big challenges with clinical trials is that you measure the performance of the patient on scales, like there's an Alzheimer's rating scale at the beginning, and maybe 2 years later, and ask how much has the patient declined. And what you really want to do is measure it much more frequently, preferably in a remote setting now with COVID, and to be able to track it. So there's a lot of work going on to try and understand what are the earliest signs of the disease and what are the indicators about the type of disease you might have. Alzheimer's Research UK has an initiative called the EDON initiative, Early Detection of Neurodegeneration. There are European initiatives working on that, one called Radar AD. This is kind of like the front, really, it's the front of research at the moment. So as well as trying to develop drugs, we also have to have the systems and the technology to know who has which kind of disease and is the drug working. So both of those arms need to be really funded and researched.
So if I put together that answer with something you said, the different types of dementia manifest themselves in different parts of the brain, as I understand it, and therefore those cognitive tests or the memory tests, by testing the behaviour of different parts of the brain, you can work out which is the underlying mechanism.
Very
yes?
simplistically, David, that is a goal. I'm not sure we're quite there yet, but that has a lot of validity to it. Okay. The thing to bear in mind, though, is it is a complex disease, and in some ways it's sort of 20 years behind cancer, in that there are many things that contribute a small amount. So you might have a not particularly useful type of gene that inclines you to frontotemporal dementia, but something else in your immune system that is more generally impactful. So in principle, what you say is right, but many things will contribute a small amount.
Okay, so the other thing you said is that there's a predisposition to dementia, but you kind of hinted about, you know, stress on the brain. I mean, you mentioned dehydration, which made me think about hangovers and things like that. But I mean, well, you've known me for too long. Is it the fact that there are bad things you can do to make you a bit more likely that you get dementia, more than there are good things you can do to put it off? But as you said, you know, more brain training stuff. But are there things you can do wrong to make it worse?
David, I think you should rest assured we don't know enough to say that's true. I don't know we can say that you can make it worse. We do have some evidence now emerging to say that if you look after your health, certainly in middle age, you can reduce your propensity. Some of the risk is modifiable. And in fact, there is emerging data that says the incidence of Alzheimer's disease might just be going down a little bit, because we now do take better care of our hearts, we take better care of our blood pressure, our lipids, all of this. And what's good for the heart is fundamentally good for the head. That's worth remembering.
Oh, okay. Okay, Will, time for the questions.
Yeah, so we have a few questions from the audience. The first one is about amyloid versus tau. So is there any known correlation between the formation of amyloid plaques and tau toxicity? And further to that, could tau versus amyloid just be different diseases? Why couldn't that be the case?
Yes, so amyloid generally precedes tau. So if you look at the time course of dementia, the amyloid plaques come in before tau, and there have been quite a lot of interesting studies done showing that amyloid might precipitate the tau problem. But the fact that tau is in different brain regions makes me think it's more directly linked to the type of disease that you have. There are some really good correlations. So at one end of the spectrum are people who have an inherited genetic disorder. There's a problem with a gene, their parent has got it, they have the same gene, they are going to get it. And that comes in both varieties. So there are 3 genes I can think of, amyloid precursor protein and 2 called presenilin 1 and presenilin 2. They are both dominant inherited genes. They're problems in the amyloid pathway. There's a tau gene that is dominantly inherited, and that gives you frontotemporal dementia only. It's only a tau problem. And they are extreme and unique. And then there's a whole assortment of people who have mixed dementias and have both tau and amyloid and other factors involved. So only the extremes are really separable. Most of us will fit somewhere in the middle where multiple genes contribute a very small amount in affecting your risk of dementia. Now, interestingly, on the genetics though, Julie Williams at Cardiff has been working on looking across the entire genome to understand which genes might have something to do with dementia. And she's done enough people now that she can calculate your genetic risk of getting dementia. It doesn't matter, you don't have to be an extreme example. She can calculate the genetic risk of getting dementia, which is accurate to about 80–85%. So interesting.
Thank you. We have another question from the audience here, which is that, do you think that poor BBB penetration of therapies is a major contributor to the clinical failures, or is it still a lack of understanding of biology, which is the biggest issue?
Well, there's somebody who really knows about something more about what I'm talking about. So I think the important point here is that for both tau and amyloid, and actually other approaches, people are using antibodies. Antibodies normally circulate in the blood and don't get terribly well into the brain. And there is a whole debate about whether the antibody removes proteins in the blood and then the equilibrium pulls them out of the brain, or whether enough antibody gets into the brain to signal, to tag the plaques and tangles and signal their destruction. So the blood-brain barrier is incredibly important. It's also important because it helps control the physiology of the brain. And if you have to have, as I was saying, more of a stressed situation, pH changes, dehydration, precipitation, you know, proteins are not in their most suitable chemical environment, that can encourage proteins to fall out of solution and then become toxic. So the blood-brain barrier is important in 2 respects. One, in the access of medicines to the brain, really important. And in many of the early studies, we didn't know enough about how those drugs got into the brain. And some of them did fail because not enough drug was getting into the brain. We know better now. And we understand a lot more about how drugs get into the brain, how much needs to get into the brain, and actually how the blood-brain barrier itself is an important component in the disease.
Thank you. So to put forward another question, of the drugs that you've talked about, which are currently in clinical trials, which, in your opinion, would be is the most effective? And to add another question onto that, do you think that in the future we could be using things like stem cell technology to address dementia?
So I don't have an opinion about which is going to be the most successful. I have no crystal ball. I wish I did. I think we just have to look at the data as it comes out. I don't have a favourite or any special insight really that any other scientist doesn't have. So I couldn't call a winner at all. And on stem cells, that's really interesting. So stem cells are absolutely brilliant at models. They're being models of neurons and even neurons with astrocytes and microglia, and understanding in a test tube the mechanisms that go awry in dementia. They're really good at creating a tool system where you can try drugs and see how the drugs work, understand the toxicity, understand what the drugs do to neuronal processes. Stem cells are brilliant for that. I think I would be more hopeful that small molecules or biologic drugs will be effective in a shorter time frame than stem cells. There's a lot we don't understand about stem cells, and to put cells into a brain which has quite a toxic environment, not very hospitable for cells, and expect the cells to function. So neurons are massively interconnected, and the stem cell, if it's to really function as a neuron particularly, has got to integrate in the network. You know, that's like, you know, trying to put I don't know, put a building in the middle of New York that's connected to all those around it. It's not a very simple thing to do. So I'm not sure that stem cells as neurons are going to be easy to do. If it's a stem cell that's just a factory producing a growth factor, or even acting more in an immune surveillance way, I could see a stem cell differentiated into those type of cells more immediately useful. But I'm expecting to see results on the small molecules and biologics before the cell therapy.
Hi, thank you. You talked a little bit about the level of the pH in the brain and how that can affect the solubility of proteins. Is it possible to change the pH in the brain in order to prevent insolubility of proteins forming plaques?
Well, so this is a very recent hypothesis, and there are quite a few companies now springing up looking at how proteins, how changes in the milieu of the brain, how different subcellular compartments in neurons handle damaged proteins. This is really very new, and there are companies now working on this, but I would say most of these are preclinical. It's a great hypothesis. It's certainly worth following up, but it's a little bit behind some of the other theories that we're working on. But it's a new and exciting idea, and it'd be good to see how it pans out.
Thank you. I'll put in another question. What does the work on exercise and diabetes connected to dementia show at the molecular level? Is blood glucose implicated in dementia?
Some people think it is, yes, and there are companies working on that. So yeah, there is an emerging hypothesis that there is a form of diabetes that is centrally very important. It's not my area of expertise, but there are people working on it. Yeah, very recent.
Okay, another question. Can you say anything about the role of alpha-synuclein aggregation in dementia? Is this less prevalent than tau tangles or amyloid plaques?
My, what an educated audience we have. I didn't even mention alpha-synuclein. More common in Parkinson's disease than in standard forms of dementia, but it is an aggregatable protein, and it's not one of the most common in dementia, but it certainly is found.
Okay, shall we go for one more, David, do you think?
Yes, one more.
Should we go for one more?
Yeah.
So energy drinks, which contain tau as an ingredient, do you think this is something that could increase the risk of Alzheimer's with long-term consumption? I know we touched a little bit upon what you could do to make it worse, but is there anything you could say specifically about tau as an ingredient, if that makes sense?
So we might be talking about different taus. So I'm not a great drinker of energy drinks, so I don't know everything that's in them, but I think it's more likely that the tau in an energy drink is taurine, which is an amino acid, and that's quite different from the tau that I've been talking about, which is a small molecule peptide that is part of the transport system of a neuron. So one is an amino acid chemical, and the other one is part of a much larger molecule, part of the transport system. So if you see tau in an energy drink and it's taurine, I mean, I can understand that. If you there would be absolutely no benefit to drinking tau, the molecule that's involved in dementia, in an energy drink. Taurine, possibly.
I guess say, don't look at me. I don't know about energy drinks either.
That's an unexpected question.
The
for which I
curveball
have
at the end of the
no
day.
answer.
Think of it in those terms. So time to draw it to a close. Thank you, Ruth, for an excellent presentation and handling the questions. But most as far as I'm concerned, I've heard things over the world, but it basically made it a lot clearer.
Thank you, David. I really appreciate that.
If you have any interest in what we do, these are the contact details for the Society of Chemical Industry. But it's just my pleasure to thank Ruth for a truly wonderful presentation. And I hope you will all watch the video when it comes up on the SCI YouTube channel in a couple of weeks when we've edited out all my bloopers. Thank you very much, Ruth. Again, just think of the virtual applause that you would be hearing if it was all right.
My pleasure. Thank you, David.
Take care, everyone. Bye.